Gosh, we know so much now, right? We know so much about neurons and how they do what they do. Recently, we were talking about EPSPs and IPSPs, and we were talking about graded potentials. And the reason it's important to understand EPSPs and IPSPs and graded potentials in general is that it is graded potentials. It's the job of the graded potentials to give information to the cell body. And then the cell body is going to basically add up all of that information and decide, should I have an action potential or should I not have an action potential? So the graded potentials start here and here, and they are basically sending information to the cell body. And the cell body is going to add it all up. Is going to add up all of the EPSPs, and the EPSPs are kind of like yes votes. Definitely, Lady Gaga died in a tragic boating accident, right? And the IPSPs are all the people that are contributing information going, no, I know someone in the Lady Gaga fan club, and that's not true, so don't share that information. And then it'll be the cell body's job to go, okay, I've added up all of the yeses and all of the nos, including the no from my friend who should know. And I'm like, no, I'm not going to have an action potential. Or yes, I will send that information along. Okay? So that's what all these EPSPs and IPSPs have been about. They are about, will this particular neuron have an action potential right now, right? So we need to talk about summation. When I say that all of that information is going into the cell and that the cell is adding it all up, I'm trying to describe summation. Summation is adding up the EPSPs and then subtracting the IPSPs. And there's another EPSP, another IPSP, right? And if this was where we were, then we would not have an action potential. It's only because there's been this extra EPSP that finally got this particular cell body to the moment of threshold. Don't memorize those things, okay? Don't memorize those numbers. But all the EPSPs and the IPSPs, they all got added up. And if that number gets to this moment of threshold here at the axon hillock, then the cell is going to go ahead and have an action potential. So threshold, that is the moment or the amount of electrical energy across the membrane where if we hit that, we start to open voltage gated channels. And when you start opening voltage gated channels, boom, you're going to have an action potential. And then this cell will have an action potential, and then it will release neurotransmitters at its axon terminus that will be captured by the next cell in line. And if this cell gets convinced, then this cell will have an action potential. Right? Okay. So summation. Now, you need to know that there are two different kinds of summation. The truth is, in living systems, it's kind of both going on at the same time. But let's talk about the two types. First, there is temporal summation. Temporal means time. And this means one neuron, just one neuron, okay, is sending out very frequent action potentials. And more action potentials reaching the end of that axon means more exocytosis of neurotransmitters, means more neurotransmitters inside of the synapse, means that that cell is going to open more of its ligand-gated channels, which is generally going to make it more likely that this cell is going to have an action potential. So it is possible for one neuron to be sending such frequent and intense action. All action potentials are the same, so I shouldn't say intense. Such frequent action potentials that it can convince that all by itself the next nerve cell in the line to go ahead and send out its own action potential. That would be temporal summation. A little more common is what is known as spatial summation, where we have got different neurons all talking to the same cell body, and all of them are saying a little bit, yes, I think you should have an action potential. And spatial means over space, so different parts of the cell are receiving neurotransmitters. The neurotransmitters are opening ligand-gated channels. The ligand-gated channels allow ions to come in. And if it lets sodium come in, that is going to be excitatory and make it more likely for this nerve cell to have its own action potential. And that's called summation. So now you know that the dendrites in the cell body, they receive information. How do they receive information? They receive information when the axon terminus of another axon, or in this case, two axons, okay? The axon terminus releases neurotransmitters and opens ligand-gated channels. And when it opens ligand-gated channels, it could have big graded potentials. And if those graded potentials are big enough or add up to enough to reach threshold here at the axon terminus, axon hillock, then this cell will have its own action potential and it'll release its own neurotransmitters to the next cell in line. So it would be correct to say that graded potentials can cause an action potential. Action potentials cause the release of neurotransmitters. Neurotransmitters cause graded potentials, and then graded potentials can cause another action potential. All right, here we've got a question. Ligand-gated channels, what's true about them? Open when the proper molecule binds to the protein. Well, that's true. That's true. May allow chloride to enter the cell. Yes. And when they allow chloride to enter the cell, you get an IPSP. And when they allow chloride to enter the cell, you get hyperpolarization. If I'm bothering to write it out, it's on the exam. They may allow sodium to enter the cell. That's true. This would be EPSPs, and EPSPs depolarize the cell. Okay, so that means they can hyperpolarize or depolarize the cells. So, all the above are true. All right. Yeah, there are different channels in different parts of the neuron, right? There are ligand-gated channels in the dendrites and cell body because ligand-gated channels create graded potentials, and this is where we get our graded potentials. There are voltage-gated channels in the axon because voltage-gated channels create action potentials, and that is where the axon happens. Where do we find sodium-potassium pumps? All over the place. Let's talk about myelin. Let's talk about myelin. Myelin is not a molecule. Myelin is a mixture of fatty molecules, and myelin is created in the peripheral nervous system by Schwann cells, or in the central nervous system by, do you remember? You've been studying, what is it? Oligodendrocytes, yeah. Okay. Now, Schwann cells in the peripheral nervous system are always found wrapped around the axons of peripheral neurons. But not all Schwann cells make myelin. There are some of your nerves, particularly some of the nerves that carry pain. There are some of those neurons that are not myelinated. And they still manage to carry an action potential quite nicely, just not as quickly as a myelinated axons. A myelinated axon, here's our Schwann cell, and as she was sitting there, she was like, oh, I think I will just roll myself around this axon many, many times. And when they do that, then they create myelin down here inside of, they create myelin down there on the inner layers. So the myelin sheath and the Schwann cell are not the same things. The myelin sheath is a part of the Schwann cell and was made by the Schwann cell, but it is the structure filled with myelin that was made by the Schwann cell. The Schwann cell has, I guess, she's got her own life too. I don't know. But let's talk about why it's so important. I already said oligodendrocytes do it in the central nervous system. By the way, this myelin stuff, it looks white. And that's why when you're looking at a brain or when you're looking at, remember the spinal cord? The spinal cord has got this like gray butterfly thing in the center. That gray butterfly area, no myelin. The stuff on the outside that looks white, myelinated. So let's look at a myelinated neuron. Now we know that the dendrites and the cell body, that's where we will get graded potentials because that is where we will find ligand-gated channels. That summation will happen, and if we reach threshold, we will start to open up voltage-gated channels in the axon hillock, and then we might get an action potential, and an action potential happens in the axon because that is where we will find voltage-gated channels. Good, I'll review. Now, this particular axon is myelinated, meaning it has got Schwann cells wrapped around it that have made myelin sheaths. Now, these myelin sheaths are not continuous. You might notice that the myelin sheath looks different on this drawing than it does on your lab model that you learned in 150. I am informed that the lab model is the more accurate representation, but it doesn't matter too much for us, except for I want you to know that the myelin sheath is definitely never contiguous all down the length of the axon. And that's not because the body couldn't do that. If the body wanted to, the body could make it so this myelin sheath, which is actually, let me just represent the myelin sheath as being orange, okay? Right now, this myelin sheath does not touch the edge of this myelin sheath. And where the area where they are not overlapping is called the node of Ranvier. The body could make it so that myelin sheaths are touching. The body deliberately does not do that because myelin sheaths are not just insulation. Yes, the purpose of a myelin sheath is to insulate regions of the axon, but it's not doing it for the same reason that we will insulate an electric cord or insulate the wires in the walls. It's not done for the same reason. Let me explain to you why we are insulating these little areas of the axon. All right, so here we've got just a really badly drawn axon, okay? So these areas where there is no myelin, actually the Schwann cells would be touching each other, so this is an even worse representation, but right there, that would be the node of Ranvier. And these areas where the beige stuff is, those are the areas that are being insulated by the myelin that's created by the Schwann cell in the myelin sheath. Myelinated axons like this one will transport their action potential more quickly, even though this neuron uses less energy. That's kind of crazy, right? Something that happens faster while using less energy. Usually faster means you have to put in more energy. Myelinated axons, it's the opposite. And we will start there at the beginning of the next video.